Hello, everyone, and welcome back to Dr. Brennan's Anatomy and Physiology podcast. Today, we're going to be talking about muscle physiology. How do muscles do what they do for the human body? And speaking of how and what muscles do for the human body, let's just go over the muscular functions for a second. As most of us already know, muscles can move the body around. If you want to bend your arm and pick something up, it's skeletal muscles that do that. If you want to even just maintain your current body position, your head needs to be held up by muscles and preventing it from falling side to side. But also if we need to move things inside and around the body, muscles are really good at doing that too. Like during respiration, when our diaphragm pulls air into our lungs, we need a muscle to move that air inside and outside. If we need to move stuff through our body, like through our GI tract, we can have muscles wrapped around those tubes that'll constrict and move stuff through those tubes. What else can they do? Obviously, when our heart contracts, it moves blood around the body, so lots of movement of stuff inside and around the body. When these muscles contract, the fibers slide past one another and they can produce body heat. So when we shiver, we could maybe heat back up. And then finally, the thing that I'm doing right now, humans have developed specialized muscles for communication. So my muscles in my tongue and mouth are shaping the air as it's coming out of my mouth, and my vocal cords are vibrating and moving to make these sounds. But even outside of just like vocal communication, we can also express and communicate through movement of the body or movement of facial expression. So lots of communication function as well. So how do muscles do all of these functions that we just talked about? Well, muscles have these 4 interesting properties that allow them to act in the way that our muscles do. And you can pause the podcast right here and see if you can already describe these 4 properties, and I'll do it for us right now. The 4 major muscle properties that give our muscles the ability to do what they do is, first, a muscle is contractible, or it has contractility. It has the ability to shorten and produce a force. So like when my biceps brachii muscle contracts, it brings my forearm closer to my shoulder. Pretty cool. And that's like the most important muscle property. It's sort of the reason that we can use muscles in the way that we do. But there's 3 other kind of interesting side related properties that I'd like us to know about. Muscles are excitable, or they have the ability to respond to a stimulus, which is really cool because not only do they contract like in a response to something, we can actually stimulate our muscles to contract whenever we want them to. Really important property, excitability. Muscles can also stretch beyond their original resting length and move back to their original length. So we can, muscles sort of can stretch beyond their normal length, and they're still able to contract at that point. And that ability is called extensibility. And then our muscles are also elastic, or they have elasticity. And this is where we can stretch a muscle and still fire it, but we can also stretch that muscle and it'll just return to its original length, which is awesome because it allows for our muscles to be pretty flexible and resilient. So those are 4 major muscle properties that I'd like us to know about. Let's chat about the histology of muscles and talk about the 3 major types of muscle tissues. So you can go ahead and pause the video here and see if you can list the 3 major types of muscle tissue and the important features of each one. And I'll go through them now. We've got skeletal muscles, smooth muscles, and cardiac muscles. 3 muscle tissues that all kind of act in similar ways. They contract and produce a force, but the way they achieve their function is a little bit different. Skeletal muscles are the ones that we're going to talk the most about, and these ones are the muscles that are attached to our bones, and they can move us around. They can move our face and allow us to provide facial expressions. When we pull air into our lungs with the diaphragm, that's another skeletal muscle. And these are really cool. They're voluntary and they're controlled by our nervous system. Again, we're going to talk the most about these skeletal muscles, so we're going to kind of leave it at that. The other 2 types of muscles that we'll talk about more in future courses and future podcasts are smooth muscles and cardiac muscles. Smooth muscles are interesting because these ones are under involuntary control, and they usually wrap around circular tube-like objects like the GI tract or organs, and these are under involuntary control. And these ones are mostly controlled by either the automatic part of our nervous system or the endocrine system, which might secrete a hormone and cause these muscles to contract. Where do we find smooth muscles? Like we said, they're around most of our hollow organs, so they can contract and move things through tubes like our GI tract or blood vessels that can constrict and allow less blood to flow through a blood vessel, or dilate and allow more blood to flow through. The muscle that's embedded in our dermis, that erector pili muscle that causes goosebumps, another example of a smooth muscle. Any other ones? Yeah, the muscles maybe around our eyes. I should say in the iris of our eyes, so the colored part of our eyes that can dilate and constrict to allow our pupils to be bigger or smaller. Those are going to be smooth muscles. These muscles are what we call autorhythmic, meaning that they kind of determine their own times that they stimulate and contract. And not all smooth muscles are autorhythmic, but a bunch of them are, meaning that they sort of have their own set pattern of constriction. Sometimes it's when they stretch too far, then they'll constrict in response to that. Cardiac muscle. This is going to be the muscle that's found in our heart, and it's the major source of the movement of blood throughout the body. Cardiac muscle is also autorhythmic, meaning that it also dictates its own rhythm, and in the circulatory unit, we'll talk a lot more about that. Okay, so those are our four major types. Was there anything else that's kind of cool about these? I think maybe the things that we want to add in to our understanding of these three major types might be keep in mind their cell shape. So skeletal muscles are these like long tubes that have multiple nuclei per cell, whereas smooth muscle have these tapered ends. They sort of look like a football almost, with these tapered ends. They're spindle cell shaped. And then heart muscles or cardiac muscles are, they're kind of cylindrical, but they have branches to them, and you can see little gaps in between them. They're striated like our skeletal muscles are, and there's usually only one nuclei per cell in cardiac muscle. And the kind of the new information that we would want to be aware of is that in our spindle cell smooth muscle tissue, we have these things called gap junctions, which allow for communication between smooth muscle cell types or smooth muscle cells, and it can oftentimes lead to one muscle contracting and causing another muscle cell right next to it to contract. And we'll talk maybe a little bit more about this when we hit the central nervous system unit. But the biggest thing that I did want to include here is that heart muscles, when they're connected end to end, they're connected at these things called intercalated discs, and they sort of bind one cell to another cell. And they also have some gap junctions that allow for flow of information between one cell and another, and contraction of one cell sort of stimulates contraction of another cell. Okay, let's chat next about skeletal muscles. So we're going to spend pretty much the rest of our time talking about skeletal muscles. And let's think about the biceps brachii, the muscle in our anterior brachial region, right in our upper arm. And when our biceps brachii contracts, it brings two bones together and it causes elbow flexion. Well, if we were to take that muscle and isolate it, pull it off of the human body, slice it the long way so that we could look at a cross section of it, we would notice a few major structures here. The structures that we would notice is it looks like muscles are made up of these bundles of bundles of bundles. So when you slice a muscle, it doesn't really look just like smooth tissue right there. Instead, we see these regular repeated groupings with smaller groupings inside of it. So we have an entire muscle, and if we slice that muscle, the first grouping that we would see inside of it are these things called fascicles. We would see a little bit of connective tissue wrapped around several or a bunch of really sort of obvious to see from the naked eye bundles of muscles, and we would call these things fascicles. So inside of a whole muscle, we would first see a bunch of fascicles. If we take that fascicle and look down its long axis and look at what is the cross section, what makes up these fascicles, we would then see muscle fibers inside of it. And as we were looking at this, we would need a microscope to see these things, but we'd see these long tubes of muscle fibers or muscle cells. Those two terms are used interchangeably. So we'd have these long muscle fibers, these long muscle cells inside of our fascicles. Oddly enough, if you take that muscle cell, slice it the long way, and look down its long axis, we would see another bundle, several bundles of things inside of this muscle fiber. And these bundles inside of a muscle fiber, these are called myofibrils. And these myofibrils make up these big bunches inside of a muscle fiber. And guess what? They also have long tube-like structures down the long axis of those. So our myofibrils have smaller tubules inside of them, and these things are called myofilaments, and there are two major kinds. There's actin and myosin that run all the way down a muscle cell. So we have actin and myosin, and those are our primary myofilaments that we're going to be looking at when we look at muscle contraction. So I'd like us to review those anatomical terms for a muscle and be ready to maybe describe the relationship between all of those. Speaking of which, while we're looking at a muscle sort of from its macro level and then zooming into the cellular and subcellular level, we can notice several layers of connective tissue wrapped around these structures. We have epimysium that wraps all the way around the muscle, and it holds a bunch of fascicles inside of it. Each fascicle is wrapped up in a connective tissue called perimysium, and then each muscle fiber, each muscle cell, is wrapped up with something called endomysium. And so these three major pieces of connective tissue are what make up these regular repeated bundles that we can see inside of a muscle. And keep in mind the epimysium, the one that's wrapped all the way around the outside, sometimes it merges straight into a really thick layer of connective tissue called fascia. And you might have heard that before, like if you have plantar fasciitis, it's this big thick piece of connective tissue on the bottom of our foot kind of wrapped around a muscle, and it can sometimes get inflamed. And that's, again, plantar fasciitis, or the inflammation of the fascia. As we move inside of the muscle cell, let's zoom way in and look at an individual muscle cell. So we have these long tube-like structures wrapped in that endomysium, and then inside of that we'd have a bunch of tubules that then hold a bunch of other structures here. So we've got these myofibrils, or these tubes inside of our muscle fiber. If we pull out these myofibrils and check out these long tubes inside of muscle cells, we would notice a regular repeated pattern all the way down the myofibril. We'd notice these regular repeated light and dark bands. And these light and dark bands are made from the actin and myosin overlapping with each other and not overlapping with each other in a regular repeated pattern all the way down the myofibril and then all the way down the muscle fiber. And these regular repeated bands we can break them up into a basic unit that we can use to describe and talk about a muscle fiber or a muscle cell, or these myofibrils inside of the cell. These regular repeated units that go all the way down the myofibrils are called sarcomeres. And what the sarcomeres are is it's where actin is attached to these end discs on the ends of these sarcomeres. And so we've got these Z discs from one end of a sarcomere to the other end of a sarcomere, and we've got actin attached directly to those Z discs, and then myosin sits in the middle and overlaps partly with some of the actin, and then it has its own space where it's only myosin. And what we can do is actin and myosin then slide over one another, and it brings one Z disc closer to the other Z disc during muscular contraction. If we look at this sarcomere, hopefully you have either like an image or you can imagine it in your head. We've got our Z discs connected to actin. our myosin or our thick filament in the middle, and then these two filaments are going to slide past one another, we can see some regular repeated zones or areas of our sarcomere. Like we said, we've got Z discs on one end and the other end, and then there'd be another sarcomere on one end of the sarcomere that we're looking at, and a sarcomere connected to the other end. And straight down the middle, we have this M line where we would have only myosin. And in that entire zone that we have only myosin down the middle, we would call that the H zone. Where we only have actin on the ends of the sarcomere, we would call that the I band. And then the A band is from one end of the myosin all the way to the other end of the myosin, which includes some area where the myosin overlaps with actin, and then an area where there's just myosin. Again, that was that H zone. So I would like us to be able to describe these different regions, and then specifically talk about how do these regions change when muscles contract. And here's the deal. When muscles contract, actin and myosin slide past one another, and it brings the Z discs, the ends of the sarcomeres, closer together. And if all of the sarcomeres have their Z discs brought closer together, it causes a pretty significant shortening of the muscle. And the muscle sort of widens out or fattens out in the middle, and that's how muscle contractions work. And as we're looking at the sarcomere, let's think about which one of these zones would shorten during muscular contraction. Well, the entire sarcomere shortens. It brings one Z disc closer to the other Z disc, so the entire sarcomere gets shorter. The I band, the region on the ends of the sarcomere that only has actin, that would also get shorter because we would be sliding the myosin closer and closer to the Z disc, and so that zone where we can only see actin on the ends, that would also get shorter. In the middle, where we can only see myosin at that H zone, that would also get shorter because again we'd be sliding the actin closer and closer to the center and causing the zone that only has myosin to be a little bit smaller. Now let's think about the A band. The A band is the region from one end of the myosin to the other end of the myosin. And as our sarcomere gets shorter, that region actually doesn't change at all because actin and myosin don't get shorter in this process. They only overlap more or less with each other. So that's like a 1-2 of our sarcomeres and the regions that are really important for us to understand. Next up, let's think about how do actin and myosin slide past one another? Well, we've just said that a common misconception about muscle contractions is that maybe actin and myosin are like these rubber bands, and they stretch out and then they can contract and cause themselves to shorten up. And that's not actually what happens. Actin and myosin don't change length during skeletal muscle contraction. Instead, what happens is the overlap between the two gets greater or smaller. So if we pull a muscle and lengthen it, we would pull those actin and myosin further apart, and they would slide across one another and be less overlap. And then if we allow the muscle to contract, actin and myosin slide past one another, and there would be more overlap between these two. You know, while we're talking about this, keep in mind that an individual muscle, it can contract with a force and shorten, but it really has no mechanism to like push itself back out to its extended length. Instead, what we would need is a different muscle that's going to produce the opposite effect and pull on that muscle and extend it back to its original length. Okay, so we've got our actin and myosin. They can slide past one another and become shorter, and here's how that process works. Myosin has these little golf club shaped heads on them that whenever they see actin, this golf club shaped head grabs onto myosin and pulls on it and causes more overlap between actin and myosin. ATP's role in this process is to bind to that myosin head and extend it back into its loaded position. So it grabs onto the myosin head, pulls it back into that loaded position, and then if it sees actin, it just grabs onto it and pulls on it again. As long as there's ATP present and myosin can see actin, it's just going to keep grabbing onto that thin filament of actin and pulling on it. So we need a mechanism to prevent this spontaneous muscle contraction. As long as there was ATP and myosin can see actin, this muscle is going to want to contract and shorten. So what we do is whenever we don't want muscles to contract, we wrap the actin in these two interesting structures called troponin and tropomyosin. Sometimes they're called troponin and tropomyosin. I call it troponin and tropomyosin. And tropomyosin is this long protein that can wind all the way around actin, and it covers up any of the active sites for myosin to bind to it and pull on it. So while tropomyosin is wrapped all the way around actin, the myosin head cannot see any active sites, and it can't bind to actin and pull on it, and we can extend a muscle, and the muscle can be in a relaxed state. But what we need a way to cover up the active site on actin sometimes when we want our muscles to relax, but then also we want myosin to be able to see actin and contract when we want it to. So here's where troponin comes into play. Troponin is like this little lock mechanism, and it locks tropomyosin in place. And whenever we want, we can unlock troponin and move tropomyosin out of the way. So here's the analogy that I like to use. If you want to lock your bike up so that it can't move out of the way, nobody can steal it, we would take a big bike chain and you'd wrap it all the way around the bike, and then you would lock that bike chain in place, and you'd have a key that can unlock it. And here's the deal. Muscle contraction is exactly the same. When we don't want muscles to move, we wrap a huge bike chain around actin, and this bike chain that we're wrapping around actin is called tropomyosin. It wraps all the way around actin, and it prevents muscles from contracting. And the lock that we put on tropomyosin and that holds it in place is called troponin. And the key that we use to unlock troponin and move tropomyosin out of the way is calcium. Whenever we flood calcium into the myofibril, calcium binds to troponin, and it unlocks our locking mechanism. Then we can move tropomyosin out of the way, and now the active site on actin is exposed, and that spontaneous muscle contraction would occur. Myosin can bind to actin and pull on it and cause muscle contraction to occur. So I would like us to be able to explain those different structures that we were just talking about. Actin and myosin, and how do muscle contractions occur with those two, and then what's the role of troponin, tropomyosin, and calcium in preventing or allowing muscle contraction to occur. Next up, let's talk about the ways that we can get ATP in the muscle in order to reload those myosin heads and allow for muscle contraction to occur. There's three major ways that our body can store this ATP or create ATP in order for muscle contractions to occur. First of all, we can undergo this thing called cellular respiration. And this is where blood glucose and a couple other things, they undergo cellular respiration, where we break down glucose and we produce a bunch of ATP in the process. And this happens, a lot in our the mitochondria, all these mitochondria that are in our skeletal muscles. They take blood glucose and a few other things. Oxygen is needed in this process, and they break down that glucose molecule, and we can exhale CO2. Water is produced in the process, a little bit of heat is produced in this process, and then we get a bunch of ATP. And all of that ATP can then go into this process of reloading those myosin heads and causing muscle contraction to occur. Our body has another way that we can generate this ATP, and that's called glycolysis. And this is where we take glucose and we snap it in half through a process called glycolysis, and we produce a little bit of ATP from this process. This process is not as efficient as cellular respiration, but it doesn't need oxygen for glycolysis to occur. Cellular respiration, we take blood glucose and we use oxygen, and we completely break down glucose into carbon dioxide, heat, water, and a bunch of ATP. Glycolysis is a little bit weirder, where we just snap glucose in half, and then we can get a little bit of ATP from this process, but we don't need to use oxygen. The downside is that we don't get as much ATP, and we get these two sort of half broken down sides of glucose, and we need to convert that into something called lactic acid. And lactic acid is just a byproduct that our body can sort of reuse, or it can sort of stick around in muscles and hang out there. So we had cellular respiration using blood glucose and oxygen to produce tons of ATP. If we don't have oxygen, we can use glycolysis, which is where blood glucose is just snapped in half and we get a little bit of ATP, not too much, and we're left over with some byproducts like lactic acid. And then finally, if we have free-floating ATP in our muscles, ATP is kind of highly reactive and it's pretty volatile. It wants to bind to stuff whenever it can. And if we let ATP just hang out too long, it will oftentimes just spontaneously react and get spent. It's like the cash in your pocket. If you hang out with cash in your pocket for too long, it just gets spent on dumb stuff. Well, what our body has evolved to do is it can take this ATP and react it with something called creatine and make a molecule called creatine phosphates. Creatine phosphate, or creatine phosphates, these molecules can then be converted back to ATP pretty easily. So if our body has a bunch of free ATP floating around, we can convert some of those to creatine phosphates that can be stored in the muscles for a little bit longer than ATP can. And this is really great because this would allow us to convert our kind of volatile and easy to spend ATP into something that's a little bit more secure and longer term. Think about like if you had cash in your pocket and you're worried about spending it on stupid stuff, you could just go load up a grocery gift card for it and say that the only thing you can spend that cash on now is just groceries. And then it'll just sit in your wallet until you really need to spend it. So those are our major ways that we can generate ATP. Okay, so next we're going to shift gears a little bit and talk about how do our neurons stimulate these skeletal muscles to contract. And we all know that our brain can send information to specific muscles and cause them to contract. And that's what allows us to do a bicep curl and flex or contract our bicep muscle and perform that elbow flexion. So how does this work? Well, our neurons send information out and to our muscles. And where a neuron meets a muscle, we would call this thing a neuromuscular junction. And that's where the end of a neuron comes really close to muscle fibers and cause those muscle fibers to stimulate. And here's what these kind of look like. At the end of an axon of a neuron, that axon would split up into multiple little axon terminals that can then go and touch a variety of different structures, including a bunch of skeletal muscle fibers. So these axon terminals can spread out and touch a couple muscle fibers, and then whenever the information travels down the axon of that neuron, it will then jump over to the muscles, and that same sort of electrical stimulus will transfer through the muscle and cause that muscle to contract. So these neuromuscular junctions, sometimes they're called NMJs for neuromuscular junctions, they have a few major pieces. There is the presynaptic terminal, and that is the very end of the neuron that is coming really close to the muscle fiber. Then there's a small little gap, and we call this little space in between the neuron and the muscle, we call it a synaptic cleft, or a little space in between the presynaptic terminal and then the postsynaptic membrane. Sometimes it's called the motor end plate. In this case, when we're talking about a neuromuscular junction, this postsynaptic membrane is the surface membrane of the muscle fiber. And so these two structures come very close to each other, but there's a little gap in between the two. And how we get information from the neuron to the muscle fiber is kind of interesting. The presynaptic terminal has these little vesicles, or these little membrane-bound compartments filled with neurotransmitters. And neurotransmitters are like tiny little chemical messengers, and it's the way that we can turn neural information from our neurons into muscular activity or causing a muscle to contract. And in the case of neuromuscular junctions, the neurotransmitter that we're talking about here is acetylcholine. acetylcholine, or ACH. So we've got these presynaptic terminals filled with these synaptic vesicles, and the neurotransmitter inside of them is acetylcholine. And when the electrical signal from a neuron flies all the way down the axon and makes it to these axon terminals, it causes these synaptic vesicles to merge with the plasma membrane of the neuron and dump their contents out into that little gap, the synaptic cleft. So we get a bunch of acetylcholine dumped in the little space in between the presynaptic terminal and the postsynaptic membrane, or that muscle fiber membrane. On the surface of that muscle fiber, we would have a bunch of receptors for acetylcholine, amongst other things. And in the presence of acetylcholine, the acetylcholine will bind to the receptors, and it will cause another electrical signal to transmit across the muscle itself. And that electrical signal will then cause calcium to get dumped into all of the myofibrils. And calcium in the presence of inside of our muscle fibers will allow for that lock and key mechanism, troponin and tropomyosin, to move out of the way, and those myosin heads will then grab onto actin and cause it to contract. So that was sort of how a motor neuron stimulates a muscle at this specific structure called a neuromuscular junction. And I'd like us to be able to talk about, like, how does this process work? Maybe we could even theorize, like, what would happen if we were to block the release of the neurotransmitter or cause the neurotransmitter to get released at different times. I'd like us to be able to think about those things there. Speaking of which, and in addition to this, I know I'm hitting us with a bunch of different structures, but let's think about the muscle fiber structure again. And given what we now know about muscular contraction and things that happen at the neuromuscular junction, what other structures are important to recognize here? First, the surface of a muscle fiber, the membrane that surrounds the outside of the cell, typically called the plasma membrane. When we're talking about a muscle fiber, a muscle cell, we would call that plasma membrane the sarcolemma, And sarco, if you ever see that prefix, just like the prefix myo typically refers to muscle, sarco typically refers to muscle as well. It's a little bit of an older term. Sarco typically means flesh or flesh-like. So when anatomists were like tearing apart organisms and looking at their flesh, most of the time we're looking at muscle, and so we would call it the sarcolemma. Inside of a muscle cell, the cytoplasm of a muscle fiber, we would typically call the sarcoplasm. So I'd like us to know those two terms as well. A muscle fiber is coated with the plasma membrane called the sarcolemma, and then the sarcoplasm would be inside of that. So when a motor neuron wants to stimulate a muscle fiber, we'd have that neuromuscular junction with a small little gap between the axon terminal and the postsynaptic membrane. In this case, we could call it the sarcolemma. It's that membrane covering a muscle fiber. And when a motor neuron stimulates that muscle fiber, we would have these acetylcholine receptors on the surface of the cell that would bind to acetylcholine, and in that presence, it would start to transmit the signal all the way across the membrane and cause calcium to get released into the myofibrils. But if you think about the structure of a muscle fiber, you'd have this big tube with a bunch of myofibrils stacked up inside of it. And so if we were to stimulate the surface of that muscle cell with acetylcholine and cause release of calcium inside the muscle, the myofibrils that are way deep inside of the muscle fiber have a difficult time reacting to that stimulus. So what we need is structures that will allow the calcium to quickly transmit all the way through the muscle fiber. And that's where our sarcoplasmic reticulum and transverse or T-tubules come from. So sarcoplasmic reticulum is this web-like structure that is wrapped around the myofibrils, which again are the tubes that are inside of the muscle fiber, and they house the actin and myosin that are going to slide past one another. And this sarcoplasmic reticulum is a modified endoplasmic reticulum, and it stores the calcium that when a muscle fiber gets stimulated, then the sarcoplasmic reticulum drops its calcium into the myofibril. When we want muscle contraction to stop, we would then pump the calcium out of the myofibrils and store it back into the sarcoplasmic reticulum. But like we said earlier, when a muscle gets stimulated from the surface of a cell, these axon terminals, the neuromuscular junction, they only kind of touch the surface of the cell. So how does that information quickly travel all the way down the muscle and get inside of the muscle fiber to all those deep myofibrils? And that's where T-tubules come from. It's sort of an inward fold of the sarcolemma, and these transverse tubules allow for the signal to transmit deeper into the myofibrils that are deep to and away from the surface of that sarcolemma. So again, sarcoplasmic reticulum holding calcium for muscle contraction, and these T-tubules are these almost like side streets that allow for the transmission of the signal deep into the myofibril. Okay, let's next chat about how do we measure muscle contraction. Like when we look at a muscle contracting, how do we use to measure it? And we can use like force strength or what do we use? So the typical way that we look at muscle contractions, the unit that we're going to measure muscle contractions in is this thing called a muscle twitch. And this is the smallest unit of muscle contraction that you can get in a muscle. And you might have heard of a muscle twitch before. You might have had muscle twitches before. You can think about like if your eye, the muscles around your eye have ever twitched, you'll get this tiny little muscle contraction and relaxation. And that's what a muscle twitch is. It's just the smallest amount of contraction that a muscle can undergo. And when a muscle contracts in this way, we can break down this muscle contraction into a few different phases. There's a lag phase at the beginning, then there's a contraction phase, and then a relaxation phase. During the lag or latent phase at the very beginning, we would see no contraction in the muscle yet. This lag or latent phase is the time that it takes from an initial stimulus for that muscle to contract. It's usually very small, just like a couple milliseconds, and this is the time it takes for your central nervous system to send information to the muscle, and that information to go across the neuromuscular junction, acetylcholine to be released, and then calcium to get released inside of the myofibrils. So this is the lag or latent phase. Then, once calcium gets released into the myofibril, troponin sees calcium, gets unlocked, tropomyosin moves out of the way, myosin heads grab onto actin and start pulling on it, and that's when we're going to start to see muscle contraction. And so if we were to measure muscle contraction in like force or tension that's generated in the muscle, like you have somebody maybe grab onto a tension reader and squeeze it, during the contraction phase, we would see tension start to increase in that muscle. And if it was just a muscle twitch, it'd be a little bit of tension that would show up on our myogram, or the thing that we're using to measure muscle contractions. Once that force has been generated, if it's just a muscle twitch, then calcium's going to get pumped out of the myofibrils. Troponin, tropomyosin bind back to actin and cover up the active site, and muscular contraction stops, and we would see the tension in that muscle start to slowly go down back to a resting stage. And that point of contraction in the muscle starting to move back down to like a normal resting state, that's called the relaxation period. And that's essentially where calcium is being pumped back into the sarcoplasmic reticulum and muscle contraction stops. And it's useful to understand these three phases because different physiological processes happen at each stage. And if people are having different muscular issues, it's useful to know maybe what stage of muscle contraction of a muscle twitch is really being affected here. If someone can't constrict their muscles, perhaps like the lag or latent phase is a problem, or maybe there's a problem with the contractile period. Or if a muscle just maintains solid tension without relaxing, maybe there's something going on with this relaxation period. So it's useful to think about muscle contraction in this way. Speaking of which, so we know that we were just saying that a muscle twitch is sort of the smallest unit of muscle contraction that we can get. If a muscle is stimulated, a muscle twitch would show up on like a myogram, and we can see a little bit of tension and then relaxation in that muscle. Let's talk about how muscles contract. And there's this interesting sort of law around muscle fiber contraction called the all or none law, or the all or none principle of muscle contraction. And that is this: if a muscle is stimulated to contract, that muscle fiber that will contract contracts as hard as it can for as long as the stimulus is on this muscle. And then when the stimulus stops, the entire muscle stops contracting and then goes to a relaxed state. If there's not enough signal to stimulate this muscle, then there would be no muscle contraction. But as soon as we hit a threshold of enough stimulus hitting that muscle, the entire muscle fiber contracts as hard as it can. There's no in between here. Either the entire muscle contracts or it doesn't contract at all. And this is the all or none principle. The way I think about it is it's like flipping on a light switch in your room. You either flip the entire light switch and all of the lights come on as high as they can, or the light switch stays off and no light occurs throughout the room. Now, if you're thinking about this and thinking, wait a minute, can't we provide different amounts of tension in a muscle? Can't you contract a muscle a little bit and contract a muscle a lot? You can. And we'll talk about how that process works. So just as a summary here, an individual muscle fiber, an individual muscle cell, if we stimulate that thing, it contracts as hard as it can, no questions asked. If we don't stimulate it, it does not contract at all. And what's interesting about this is that a single neuron has multiple axon terminals or branches that come out and can touch multiple muscle fibers. And all of the muscle fibers that are stimulated from one neuron touching multiple fibers, that structure is called a motor unit. A motor unit is one nerve or one neuron sending information to a muscle, spreading out and touching multiple different muscle fibers. Whenever that neuron sends information to a muscle, all of those fibers will contract in one single unit. So what we can do here is each muscle has a range of a few motor units to a ton of motor units. So let's take our biceps brachii, for example, and let's make it really simple. Let's say that we split all of the fibers up in that biceps brachii into 3 groups. One neuron stimulates group A, another neuron stimulates group B, and another neuron stimulates group C. Well, now, whenever neuron A stimulates that group, all of those muscle fibers would contract as hard as they can, but some of the muscle fibers that are next to it remain relaxed, and we would get maybe a lower force generation than if the entire muscle were to contract. So if you try to pick something up and it's super light, we only might stimulate motor unit A and all of the fibers connected to that neuron, and we would produce a little bit of force. If you try to pick something up, it's a little bit heavier than you thought, and you're like, I can't really pick it up. You might need to stimulate an additional motor unit that will start firing even more muscle fibers. Now you might have enough force to pick that thing up. But if it's still super heavy, you might need to recruit motor unit C to stimulate all of their fibers, and then you might actually be able to pick the thing up. And this idea of starting off with one or a few motor units, seeing if you have enough force, and slowly recruiting more and more motor units with more and more muscle fibers in them is the way that we can get what's called a graded response of the muscle, or the muscle to contract a little bit sometimes, a medium amount in other times, or its maximum contractile strength in some cases. And this idea is called motor recruitment. Motor unit recruitment is where we take one motor unit and stimulate just those fibers, and if we need more muscle contraction, if we need more strength or more tension in the muscle, we can slowly add more motor units and make the contractile strength go up. You might have even experienced this before, like if you're working out at a gym and you try to lift something up and it's way heavier than you thought, you might not be able to lift it up right away, but then if you get down, really focus on it and lift with all of your force and activate all of the motor units, then you can pick the thing up. So motor unit or multiple motor unit recruitment is one way that we can generate different amounts of strength in a muscle. The other way that we can generate different amounts of strength is this idea called wave summation. Wave summation. And that is where, if we send information to a muscle and cause it to contract, the smallest unit of muscle contraction is a muscle twitch. So we allow that muscle to twitch, some tension increases in that muscle fiber, and then we stop sending the signal and the tension goes back down. But if we send a signal and cause a muscle to twitch, but before that muscle completely relaxes and goes all the way back to its zero tension resting point, if we send another signal right behind it, we can cause that tension to build on itself and be a little bit higher than the tension of one muscle twitch alone. And if we keep sending a signal behind if we keep sending multiple signals behind each other to a muscle, we can get these muscle twitches to stack up on one another and have an overall higher tension than the original muscle twitch. And this stacking of signals and stacking of tension in a muscle is called wave summation. Because when you look at a myogram of this muscle twitches stacking up on one another, it looks like a slow up and down wave moving up at each interval. And we might have experienced this before. If you hold on to something that's really heavy, we'll pick up like a bicep curl and sort of hold our elbow at an L and see how long we can hold on to a really heavy thing. After a while, your muscle is going to start to shake. And that is where we can see the buildup of these signals, and we see the relaxation between the signals that are being sent to the muscle. So as the stimulus keeps getting sent to the muscle, it'll contract and relax really quickly, and you might see your muscles start to shake. And this idea of our muscles shaking or a little bit of relaxation in between contractions has everything to do with this idea called tetanus. Tetanus is where we can measure the amount of, or like the smoothness of a muscle contraction. And when a muscle is completely contracted at its highest amount, and the tension remains solid, we would say that that's complete tetanus. Whereas if you hold on to something and allow the muscle to relax in between and start to like shake, that's called incomplete tetanus. And this happens in a gym if you're working out and you put an extreme amount of stress on a muscle over and over again. Oftentimes we send an incomplete tetanus signal to that muscle, and it'll shake because it needs time to sort of store back up that information that can get sent. So again, two major ways that we can have a difference in tension in a muscle, even though muscles exhibit our all or none principle. One, we could recruit different amounts of motor units to cause that muscle to contract, and a lower amount of motor units causes a lower amount of tension, and you can slowly add more and more motor units and more muscle fibers to cause a higher tension. Or we can use wave summation, which is to send a signal to the muscle at a really quick interval and cause the stacking up of those muscle twitches. As we zoom out more, we're going to look at muscle contraction from a more macro basis, and let's now look at the different ways that a muscle can contract just from the outside looking at a muscle with the naked eye. First, we can have what are called isometric muscle contractions. Isometric muscle contractions are where we put tension in a muscle, but we don't actually move the bones that the muscle is attached to. So we're putting tension in a muscle without changing the length of the muscle. You can imagine activities that require isometric muscle contraction. It would be like a plank, where you might get into like a push-up position or on your forearms, and you would constrict or contract your abdominal muscles to try and hold you up in a plank position. Those muscles ideally aren't going to be changing length, but they're going to be under tension and sort of holding us in one place. Another time that you'd need to use isometric muscle contractions, maybe you need to push on an accelerator in a car, but hold it at a steady speed. So maybe like an occupational therapist might need to work with somebody on an isometric muscle contraction where they're pushing on a pedal and holding it in place to try to maintain a constant speed. These are called isometric muscle contractions. Isotonic muscle contractions are where we put tension in a muscle and the length of the muscle changes. And there's two specific types of isotonic muscle contractions that I would like us to be aware of: concentric muscle contractions and eccentric muscle contractions. Concentric muscle contractions are where we put tension in a muscle and the muscle overcomes the resistance and shortens. This would be like the upward phase of a bicep curl. We'd hold a dumbbell in our hand, we would put tension in the biceps brachii, we would overcome that tension, there'd be contraction of the muscle and it would shorten, and the barbell or the dumbbell would come up and we'd be in our finished bicep curl position. Eccentric muscle contractions are when we put tension in a muscle, but we allow the muscle to lengthen. This would be if we were to start at our top position of our bicep curl, that finished position, and then slowly extend our muscle and allow the dumbbell to drop back down to the bottom. That would be an eccentric muscle contraction. Let's next talk about changes in muscle size. So when we put our muscles through a set of exercises over a longer period of time, we'll notice that the size and shape of the muscle might change a little bit. And so let's talk about how that happens. Muscle hypertrophy or hypertrophy is where a muscle increases in size after it's been contracting and relaxing for a long period of time. So if you go to the gym and you work out your muscles, you'll notice that those muscles will get thicker or wider in diameter, and they will hopefully be able to produce more force output over time. This process is called muscle hypertrophy, and it's how muscles increase in size. And what's interesting about them is some people probably think that as a muscle gets bigger in size, the number of cells inside that muscle must also increase in size. We must be just growing more muscle fibers. And that isn't exactly what happens. If you take a smaller muscle, work it out a bunch, and then we have a larger muscle afterwards, if we slice the before and after muscle and look at how many muscle fibers are in it, they're the same amount of muscle fibers. So what's going on here? Well, what happens is inside of the muscle fibers themselves, the muscle fibers stay the same, but the myofibrils that are inside of each muscle fiber increase in diameter and sometimes increase in total amount. We'd get more nuclei in our muscle fibers, so more nuclei would show up. And that's great because that can help make us ribosomes that are going to help us build protein. And overall, the larger muscles have a few interesting things going for them. There's better coordination of those muscles. The motor units that we need to recruit to cause the muscle to contract usually can contract faster or can contract in a more coordinated manner. There's increased number of metabolic enzymes, there's better circulation, there's usually less restriction by subcutaneous fat that's in that area. So overall, muscle hypertrophy has a bunch of interesting features that happen. If we take a really big muscle that's hypertrophed or has increased in size, and we don't work that muscle out very much, we'll notice that that muscle will decrease in size. And when a muscle goes from an increased state to a decreased muscle size state, we would call that process muscle atrophy. And muscle atrophy can happen for a variety of reasons. It might just be somebody stops working out for a long time, the muscle might atrophy, or maybe somebody's in a cast and they can't move their muscles, and we'll notice significant muscle atrophy in that way. Muscle atrophy occurs in pretty much the opposite way that muscle hypertrophy occurs. We would see a decrease in the size and number of myofibrils, decrease in number of nuclei, reduction in strength and coordination. So somebody that maybe had a cast on their arm for a couple months, maybe they'll have a tougher time doing the same activities like they used to do. They might not be able to throw with as much coordination as they used to. We'll see a decrease in circulation, possibly an increase in that subcutaneous fat that's right around the muscle. So muscle hypertrophy and atrophy, I'd like us to be able to discuss the differences between those two things. Okay, I think one of the last things that we want to talk about here are the specific types of muscle fibers that we can find inside of a muscle. And there's two broad categories of muscle fiber types. There are slow twitch muscle fibers and fast twitch muscle fibers. These are sometimes called type 1 and type 2 muscle fibers. They're also called anaerobic and aerobic muscle fibers sometimes. I'm going to call them slow and fast twitch muscles. And literally, if we were to slice open a muscle, we're going to notice a combination of slow twitch and fast twitch muscle fibers. And the distribution of how much slow twitch and fast twitch muscle fibers you have is sort of just dictated by genetics. When you're born, you have this distribution. And here's what these two types of muscles what they do. Slow twitch muscle fibers, these guys contract more slowly. They're usually smaller. They have a really good blood supply, tons of mitochondria, and these ones are really fatigue resistant. Slow twitch muscle fibers are really good at prolonged activity over a really long time. We're talking like minutes, tens of minutes, even hours of contraction. They're slower, they don't produce as much force, but they're really good at long sustained muscle contractions. So this might be like postural muscles in our neck that you can walk around all day holding our head up on our neck, moving around, and that muscle really doesn't get tired, it doesn't get sore, and there's no issue with really long muscle contractions. Fast twitch muscle fibers, though, these ones are kind of the opposite. These ones respond really fast to nervous stimulation. They can break down ATP really fast. They have a little bit less blood supply, and they don't require as much oxygen for this process to happen. They have fewer mitochondria, and when these guys contract, they can only undergo sustained muscle contraction for a short period of time. We're talking like seconds, tens of seconds to maybe a couple minutes. And there's some exceptions to this, but it's pretty difficult to get fast twitch muscle fibers to contract for longer than just a few minutes. Fast twitch muscle fibers undergo the anaerobic form of ATP production. They mostly go through glycolysis, and they just snap glucose in half and produce the ATP needed for muscle contraction. Whereas slow twitch muscle fibers, they're way better at generating ATP through cellular respiration, which does require oxygen, but it produces a ton more ATP. It's a little bit slower, but it can produce a sustained muscle contraction. So what I would like us to do is be able to think about different activities and think about, would slow twitch or fast twitch muscle fibers be more suited for this activity? So let's think about maybe jumping as high as you can, like a box jump, to see how high you can jump, or just standing and trying to measure a vertical jump height. Which muscle type would be better at doing that? Fast twitch muscle fibers would be way better at a single sustained explosive activity like jumping as high as you can. Imagine if you jumped 5 times as high as you could. Your quads and your calves would get really worn down very quickly. I doubt you could jump 5 times as high as you could without experiencing a little bit of muscle fatigue, and we see a reduction in overall force output. What about if we were to measure someone running a marathon? Would fast twitch or slow twitch muscle fibers be more adapted to like a marathon runner? Slow twitch muscle fibers would probably be better in that case, because those ones can have sustained slower contractions. We're not needing to produce a ton of force all at once. Instead, we might need to produce force in a slower amount, but over the course of like several hours. Now, the only caveat to these two things is that some people can train their fast twitch muscle fibers to act more like slow twitch muscle fibers. This takes weeks or months of training, but sometimes like cyclists can take the fast twitch muscle fibers in their quads and train them so that they can cycle for hours, and their slow twitch muscle fibers are really good at that, and also their fast twitch muscle fibers can kind of do that too. And you'll see like the veins and the blood vessels in their muscles will get larger, and they'll allow for more blood flow to that area. So I would like us to understand the difference between fast and slow twitch muscle fibers. Finally, let's talk about muscle fatigue. How our muscles end up not being able to produce the same force output over time. So muscle fatigue can be described as like a decrease in the ability of a muscle to produce a certain amount of force. So if you did 10 bicep curls After a while, your muscle would start to fatigue, or it would decrease the amount of force that it could output. So if you can lift a 25-pound dumbbell 10 times and you can't get the 11th one up, maybe if you grabbed a 15-pound weight, now you could get a couple more of those up, and you'd be noticing that the muscle is fatiguing or not being able to produce the same amount of force output. And there's a few broad categories of muscle fatigue that I'd like us to be aware of. There is something called muscular fatigue. Muscular fatigue results in or comes from a local ATP depletion in the muscle. And when ATP gets depleted, there's nothing there to pull the myosin heads back into place, and we'll notice that overall there's a decrease in force output. And when it's by a local ATP depletion, we'd call that muscular fatigue. There's a different kind of fatigue, though, called synaptic fatigue. And synaptic fatigue is not due to ATP loss or ATP depletion in the muscle. Instead, it's due to acetylcholine depletion at the neuromuscular junction, at that little synaptic cleft. If we were to try to stimulate our muscles over and over again, occasionally we would run out of the acetylcholine needed to release from the motor neuron onto the surface of the muscle cell and cause muscle contraction to occur. And when that happens, we'd say that is synaptic fatigue. So sometimes people feel fine and they feel like they should be able to lift more weight, but it's at the end of a really long day, and they're like, man, I'm fatigued even though I haven't worked out at all. I just noticed that my muscles aren't working as well as they would. That could be due to synaptic fatigue or depletion of acetylcholine at the neuromuscular junction. Finally, I saved the most interesting one for last. Finally, some people experience, and we can document and measure this phenomenon called psychological fatigue. Psychological fatigue. And this is where somehow the emotional state of the person has an ability to reduce the force output in a muscle. And it's kind of weird, but some people who experience anxiety, stress, depression, they'll notice that their muscles aren't as tuned to working as when they're not stressed, anxious, or depressed. I'll give you an example of this. I used to play tennis, and when I had no one watching me, I could hit my serve really fast, like 120 miles an hour. But when there's a huge crowd watching me and I'm a little bit nervous, my muscles are on edge, I've got some tension, some stress, some anxiety, my muscles literally won't produce that same output as I could in practice. And I'd only be able to hit my serve like 100, 110 miles per hour. And so somehow the mental and emotional state of individuals can affect their muscular output. The other time that this really occurs is during clinical depression. When we have clinically depressed patients, sometimes they literally cannot output the same muscle force as they would without that clinical depression. So you'll find that these patients have a real difficulty like just getting out of bed sometimes, or taking a shower, or having the normal daily activity. To them, it literally feels like they're lifting up 10-pound weights on their arms just to do all these activities. So those are the three major types of muscular fatigue: psychological, muscular, and synaptic. And I'd like us to be able to explain those differences. All right, everyone, good luck studying. That is what a good place for us to stop talking about muscle physiology, and I will see you soon.